In this video, we're going to be reviewing Unit 3, Cellular Energetics, particularly focusing on cellular respiration. My name is Mr. Chipman. I'm the AP Biology teacher at Murray High School in Murray, Kentucky, and hopefully this video will be a help to you. If it is, please like and subscribe, comment down below, tell me if it helped or not. Let me know if you have questions. Ask those questions in the comments below. Let's begin. Cellular respiration is about breaking down that stored energy that was made in photosynthesis, that glucose molecule, and getting that energy back out. That energy will be made in the form of ATP. The purpose of cellular respiration is to break down glucose in the presence of oxygen to make ATP. CO2 is going to be a byproduct. H2O is also being made. And so this is the exact opposite of photosynthesis. Make note of that. These two reactions are coupled, and you will see them a lot. Seems like half the tests over the last 11 years have had a picture like this, or a mitochondria and a chloroplast next to each other, or something. And they're going to ask you questions about the couplings of these two reactions. So make sure that you just understand these two equations. That's going to take you a lot of the way in understanding these reactions. A lot of times they are overanalyzed at this level, and they don't need to be. Just understanding these two equations, understanding the forest, leave out the trees, and I think you'll be good. Well, let's talk a little bit about those trees, though. Again, cellular respiration is these three steps, or not three steps here, just two. Glycolysis is the first step of cellular respiration. Glycolysis is something that is common to all living things. All living things can demonstrate some level of common ancestry because they all have glycolysis. Some of them don't need oxygen. They continue anaerobic respiration. We're able to do that as well. We'll talk about that in a moment. And those that have mitochondria, they will go on to do the next two steps in the mitochondria. Those that do not will just do it on their cell membranes, things like bacteria and others. Let's talk about those three steps. Here they are. There's three steps to cellular respiration: glycolysis, Krebs cycle, and the electron transport chain, or ETC. Glycolysis, the purpose of that is breaking down glucose in order to make pyruvate. Oxygen is not necessary for this. Glycolysis is going to continue to happen regardless if there's oxygen present or not. Some organisms do not want oxygen, and so they're going to do this anyway. Notice some ATP is being made. Some electron carriers are being made. But the main purpose behind glycolysis is to break down glyco, or glucose. And so two pyruvates are being made. If you want to put a little 2 above that, you can do that. And let's move on. If oxygen is present, well, if oxygen is not present, something else is going to happen. We'll talk about that later. But if oxygen is present, these pyruvates are going to move into the matrix of the mitochondria, and they're going to be broken down. The purpose of the Krebs cycle is to make electron carriers. Some ATP is being made, but it's very little in comparison to the whole. And so Krebs cycle, main purpose is to break down electron carriers, or break down pyruvate to make electron carriers, and those electron carriers, along with the ones that are made in glycolysis, are going to move to the electron transport chain, which is found on the inner membrane of the mitochondria. In this particular step, oxidative phosphorylation is going to occur. This is different than substrate-level phosphorylation, which has to do with enzymes. In this particular case, oxygen is going to be there to receive those electrons. Remember long ago when water was split and electrons and oxygen went their separate ways? Well, now they're being reunited, and it feels so good. And they're going to make water instead of water being broken down. It's almost like the exact opposite reaction of photosynthesis. Lots of ATP is going to be made here comparatively. Let's move on. Let's talk about the ETC. Here's the ETC. There's lots of these protons. All these proteins have names. All this is crazy. But I want to talk about a couple of these things. NADH, FADH2 are going to be the superstars in this reaction. Those are electron carriers. What do? They carry electrons. They're going to carry them, and they're going to drop them off these proteins. These proteins are proton pumps. Remember, proton pumps, active transport. They're going to take these protons that are found in the matrix of the mitochondria. Where are they coming from? They're just there. And they're going to be pumped into the inner membrane space, building up a concentration gradient. And then here's our superstar again, ATP synthase. It's going to use that concentration gradient to make ATP. And oxygen is here to pick up those electrons at the end of the long, tired day. Where do these electrons originate? In a magnesium far, far away. And here they are being picked up by the oxygen. And those two electrons, two hydrogens, an O2 going to make an H2O, and H2O is going to be found there. H2O is the byproduct of this particular reaction. CO2, the byproduct of Krebs cycle. And when you exhale both of those things. Let's move on. Oxidative phosphorylation, kind of mentioned that. We're not going to go into this anymore. Big idea, oxidative oxygen phosphorylation, something about a phosphate. Where do we, oh well, we're going to add a phosphate to make an ATP. So we're phosphorylating ADP to make ATP. Energy is necessary to do that. Where's the energy come from? The electrons that NADH and FADH2 are dropping off. Moving on. Let's talk about uncoupling because it was in the AP stuff this year. Weird. Okay, so what is this? This is basically sometimes these extra protons that are built up in the inner membrane space are going to be released back into the matrix using that gradient in order to just release heat. This can be used by an organism in order to preserve life, perhaps maybe during the winter months or something along those lines. It's kind of an odd addition to the AP curriculum, but just know when you see uncoupling, just think extra protons being used to make heat, and we're good. Moving on. Glycolysis. Let's talk about these steps a little bit more in depth. Glycolysis, break it down to make pyruvate. In the middle here is G3P, which you don't need to know. I just want to connect that back to photosynthesis, talking about G3P. There's nothing there specific, but a couple of ATP are going to be made. Sometimes you'll see the idea that 2 ATP are made twice, so you have 4 ATP, but it costs 2 ATP in order to make, in order to break that glucose. And so sometimes you'll see it like where it talks about the payoff phase. The investment phase is the first portion of glycolysis, and the payoff is the second portion. Haven't really seen any of that in the literature, so not all that important. Just understand a couple things about glycolysis. Again, it's common to all living things, and that breaking down glucose to make pyruvates. Pyruvate oxidation. If there's oxygen present, that pyruvate is going, this is one pyruvate. There's 2 pyruvates that are made. So think about this happening twice per glucose, but let's just go to the ones. So here's this pyruvate. It's going to sneak into the mitochondria. It's going to need a transport protein in order to do this, and it's going to start to break apart. One of those carbons is going to break off. CO2 is going to be given off. Anytime carbon's broken off in this process, CO2 is going to be that carbon that's given off. NADH is made, that's going to head it onto the ETC, and you have this thing called acetyl that is left, and it's going to combine with coenzyme A to make acetyl-CoA. Not a lot of this in the literature this time, but this is the thing that's going to be jumping onto the Krebs cycle, just these 2 carbons. And here's the Krebs cycle. A bunch of crazy stuff. We're not going to, look at that. Look, he's just overwhelmed. And you don't need to be, because our version of the Krebs cycle is going to be simpler. It's like this. All right? Big idea for the Krebs cycle. There's your acetyl-CoA that's only got 2 carbons in it. Those 2 carbons are going to jump on the Krebs cycle merry-go-round, and they're going to jump off in the form of CO2. You know that when you exhale, there's CO2 in it. And because of that, there's lots of things that are being made. Notice the main purpose of the Krebs cycle is to break down pyruvate to make NADH and FADH2 electron carriers. That's exactly what's happening. Right? And so that was the big idea there. Krebs cycle's over. NADH is, notice there's 4 here. Well, how are we going to make all this ATP? We're going to make it here. Each one of these NADHs turns on 3 proton pumps. 1, 2, 3, those electrons. That's going to pump 1, 2, 3 protons. 1, 2, 3 protons are going to go through that ATP synthase, and it's going to make 1, 2, 3 ATP. So each NADH that's made is worth 3 ATP. If you make a couple in glycolysis, if you make 4 times 2 in Krebs cycle, all of a sudden we have like 30 ATP that we've made. And so that is a big deal. You add 2 each for here, and you add a couple for the others that we've talked about, and you get 36 to 38 total ATP that are made in this process. Big deal. What if there's not oxygen present? Well, if there's no oxygen present, there's going to be a process called fermentation. Big idea behind fermentation is, of course, no oxygen present. That's what happens. The big idea here, fermentation happens so that glycolysis can continue. I'm going to add a little bit to that definition. Fermentation frees up electron carriers so that glycolysis can continue. What do I mean? If there's no oxygen, where's NADH going to drop off its electrons? It can't, right? Because oxygen is the final electron acceptor in cellular respiration. And so if there's none of this, then NADH is like, what do I do with these electrons? I'm just going to keep building up. And if there's if NADH can't get rid of its electrons, then no NAD plus is getting made, right? Because NAD plus is the thing that it is made after it drops off its electrons. And so if there's no NAD+, then it's one of the reactants of this reaction, then glycolysis can't happen. And so what fermentation does is it takes those electrons off of NADH so that NAD+ can go feed itself back into glycolysis and allow this to happen. This is not a good reaction. Only 2 total ATP are going to be made here as opposed to 36 to 38 that we're used to making. And so we can't live on this reaction. We will die pretty quickly. But some organisms are able to live doing glycolysis exclusively and continue to make things like lactate, which is lactic acid, which is important for the formation of cheese. Do I have one for ethanol? I don't. There's another type of fermentation called ethanol fermentation, which ethanol is made and CO2. This is like yeast does this sort of thing. And so big idea behind fermentation happens so that glycolysis continue. And this just shows the ATP to ADP to ATP idea, just that those reactions are coupled. I think we've went over that exclusively. Hopefully this was helpful. If it was, please like the video. Please subscribe down below. Please comment to tell me it was helpful. And good luck on the exam.